for this year. The Myeloid Network was founded in 2020 by our founders, as you see scrolling through, Dr. Guerrero, Varner, Gavrilovich, Johanna Joyce, and Lisa Cousins. And each month we present a topic on myeloid cells and how we can advance myeloid therapies. As you can see, we have a list of sponsors that also circulates through the slide deck. And today we have our next speaker for this month is Dr. Clayton Yates. Dr. Clayton Yates is the John R. Lewis Endowed Professor of Pathology, Oncology, and Urology here at Johns Hopkins University School of Medicine. He is the director of the Translational Health Disparities and Global Health Equity Research. He's a program co-leader for cancer genetics and epigenetics within our cancer center, the Sidney Kimmel Comprehensive Cancer Center. Dr. Clayton Yates earned his bachelor and master's degree at Tuskegee University and then went on to earn his Ph.D. from the University of Pittsburgh School of Medicine and Molecular Pathology, as well as completed a certificate of training in tissue engineering and regenerative medicine. He went on to complete a postdoctoral fellowship at Emory University with the late great Leland Chung in the Department of Urology. Dr. Yates' lab specifically studies cancer health disparities in breast and prostate cancer, and using signature molecules, they've been able to identify novel therapeutic targets and immune cells, particularly TAMs, as well as how they are associated with increased aggressiveness in specific tumor types. Dr. Yates has published robustly, Science Translational Medicine, Nature, Cell, and the like. He is the recipient of lots of federal grants, particularly T32s, U54s, as well as the Impact Award from the Department of Defense. Today, Dr. Yates will talk to us about epigenetic and genetic alterations that can contribute to aggressive cancers. Dr. Yates, I'll stop the share and let you share your screen. It's great to have you on the call this morning.
Well, thank you very much, Jelani, and I'll say I'm very excited that I have joined you here at Johns Hopkins. I knew Jelani for many, many years, and yeah, I was at Tuskegee University, and when I was deciding to come, he and I believe Otis Brawley is on the line as well, were instrumental in me finally making a decision, and I'm happy to be his neighbor as far as office space as well as our labs. And so, some of the work I'm going to talk about today is sort of looking at our journey through health disparities and how it sort of back-ended me into immunology, and particularly myeloid macrophage-associated biology. So if you can just kind of bear with us as I kind of go through how we've sort of started our focus and why we think myeloid cells, particularly macrophages, are really important for disparities. So here are a few disclosures that we have here. So if we think about, cancer rates, as Jelani mentioned, our lab has been focused on prostate and breast cancer, although we do have a current interest in pancreatic cancer, and I'll show you some data toward the end of our talk. But we kind of focus on those because of the outcomes, right? African-American men have more incidence and worse outcomes. And that is also associated with breast cancer patients as well. In fact, if you look at the etiology of breast cancer, it used to be that there was an increased incidence in European ancestry individuals, but now African-American women have actually caught up, sort of equal, and there's definitely an increased mortality among African-American. So when we start to think about looking at these rates, it's not just associated with African-Americans. We look at global rates and prostate, and sorry, I don't have the breast cancer slides up as well, but the data is almost similar. Regardless of the incidence, and there are countries where there's largely Black individuals, the outcomes are worse, right? And it sort of begs us to sort of think about the similarities that we think that could be there, particularly if we want to think about tumor biology that could be shared. As we all know, the transatlantic slave trade or the African diaspora is a route in which not only we talk about, sort of the history of the U.S., but we also talk about genetic traits sort of flowing through from West Africa to the U.S. as well as South Africa and the Caribbean. And so, early on, Ike Powell, who is a urologist at Caramos, Wayne State University Cancer Center, and he did one of, I think, some of the first studies asking the question of whether there were biological differences between African Americans and what back then it was microarrays. And what he saw was that most of the genes that were differentially expressed, again, these were at times where people didn't even understand, like, is there some differences? Is there some biological contribution to the etiology of the disease? And what he saw was there was increased inflammatory response. Interleukins, IL-8s, IL-6s, things that were upregulated in African-American men seem to be driving the disease. And if you look at other folks such as Renee Reams at FAMU University, and then Stefan Alms followed up these studies, and now I think there's an explosion of data that this data has to have been repeated. I can see at least another 8 to 10 times across multiple manuscripts, including ourselves, that the major differences that we see are immunological or innate sort of immunological response that seems to be a difference between African American and European. So, we asked the question, if this is really associated with ancestry, then, we should be able to trace this through the diaspora, right? And if you think about the transatlantic ancestry in the diaspora, if there is truly genetic consequences of this disease, we should be able to trace that. And so through the Prostate Cancer Transatlantic Consortium, which was led by Falakio Dedna, and I co-lead it with Rotimi Solomon, we started recruiting Native African individuals and comparing that to African American men. And so here's the map. So if you think about, that West African through that diaspora image that I showed earlier in my introduction, you start to see that these are the sites that we sort of intentionally were able to start recruiting from, largely based in Nigeria. And we say CAPTC, that's our consortium, Prostate Cancer Transatlantic Consortium. But now we've extended to breast cancer as well as pancreatic disease. But you can also see we not only are collecting samples, we're building infrastructure. So these are now facilities that are able to do research, they're able to collect specimens, and they're actually doing their own research. And so a part of the data I'll show you there is also really proud about the infrastructure that we are building in West Africa. So when we start thinking about some of the biology, again, we ask that question, is this inflammatory immune response that people were seeing in tumor, is that ancestry associated? So UDEL allows in a very large cohort of about 1,500 individuals, 650 from West Africa, right? And 370 plus men and African-American and sort of similar and European men in the US. We asked from a serum perspective, right, running immunoprofiling, utilizing the OLEAG assay. I'm sure many of you are familiar with that. We asked, was it an inflammatory enriched response? And what we saw was that there was out of multiple pathways that could be associated with this sort of panel, it was a 92 marker panel of proteins, we saw tumor suppression, this sort of suppressive immunity modulatory response seemed to be the most differentially expressed or most enriched in African, Native African, African American, as well, and then most lower in European. And we started to look at driver molecules that proteins that could be associated, that started to see things that could be associated with myeloid responses, right? Macrophage recruitment protein 2, also 1 is there. I'll show some data about that later, but all of the CXL, chemokines and cytokines were all upregulated as well. And so particularly in our African and Native African. So that got us to sort of thinking, like, what is this associated with? And so, again, thinking about having a cohort of individuals from West Africa and having them enriched for West African ancestry, all because they're Native Africans, we were able to sort of start to tease out, right, this sort of panel of which markers were associated with ancestry and which ones were not associated with ancestry, even if they were differentially expressed. And to our surprise, we started to paint a picture. CXL5 was one of the most associated ancestry markers that were elevated in our West African and African American, but you also saw these macrophage recruitment proteins 1 and 2 were modestly associated with West African ancestry, although they were elevated in both African and our African American men. And again, we wanted to make sure this was clinically relevant. And so if you sort of look at all the other pathways, vascular score, promoting tumor immunity, chemotaxis, autophagy, apoptosis score, and these are all the grouping from a panel. Suppressive tumor immunity score was the only one associated with worse outcome. And if you start to look at, some of the drivers of this, and this is PTN and another, regulator of Tregs, TNSFR, SF9, these markers were the only predictors of outcome in African American men. So what I'm saying to you is that with 90+% accuracy, we can predict survival outcome of African American men, and these markers did not work in European. Okay, and they only work in sort of in African ancestry individuals. So, along with this, and there's some other data that we started to publish around genomic factors, and I'm not going to present that data today because I want to really get into our sort of myeloid work. But we started to form this sort of framework, right? If we start thinking about biology, ancestry, genomics, transcriptomics, epigenomics, we got to start thinking about this from a personalized approach, but also from an ancestry diaspora approach. And so we proposed this sort of diagram that's going to be the basis of the data that I'm going to show you for the rest of our talk. And this is kind of how our lab sort of envisions approaching cancer disparities. So we assembled a large cohort of African-American men, African-American, Native African. Again, that first cohort was largely, blood-based because we were looking at serum. A number of those patients we were able to collect actual tissue from, and then we started approaching this from a multi-omic perspective, right? Running for whole transcriptomics, epigenomics, method sequencing analysis, which I'm not going to show today, and also spatial resolution and then single-cell sequencing. We also wanted to, be consistent with our ancestry analysis, and Israel Hassan, who's a graduate student in my lab at Tuskegee and now moved to me at Johns Hopkins, wanted to go beyond just running super ancestry. She actually wanted to run subpopulation ancestry analysis because she was like, there could be also contributions of, different ancestry within tribes and within Africa, within our West African populations that also could be contributing to this possible inflammatory response. And so, again, this is the framework in which we do our patients. And so when we run transcriptomic analysis of these individuals, we start to look at our self-reported ratios just here at the top. So this is Nigerian, African-American men, as far as European. When we run qualified genomic ancestry, you start to separate the self-reported race versus the ancestry. And so obviously our African-American men, 7 of those individuals who had lower than 70% West African ancestry actually cluster were European. And then there was one European patient who had over 70% West African ancestry. They were self-reported as European, but they actually cluster with African American. And so this is sort of the cluster analysis that you can sort of that is associated with this, discordance between self-reported race and ancestry. And so when we start to look at the biology of these individuals, again, counting for self-reported, which is African-American, which you see at our top of our heat map, and then subpopulation analysis, which most of our patients were from the Yoruba population, we then grouped our African-Americans and our Europeans. What was to our surprise was that although we saw the suppressive score in our serum analysis, in the tumor samples, it was quite different. So if you look at our African-American patients here, we see this clear chronic inflammatory response, which is associated with upregulation of interferon genes. Again, this is a classic signature that most people have shown. We did not see that in our West African individuals, right? And it was really striking to us. And then if you start to look at, again, this sort of, shared gene expression or the ones enriched, this inflammatory response so to be persistent in our African-American population. But when we start looking again across all, immune score, African-American men actually had immune score as well as inflammatory profiles that we absolutely did not see in our Nigerian cohort. But then when we start looking at shared genomic analysis, like what could be shared between our African-American men as well as European, what we did, and we took our bulk sequencing data and ran deconvolution analysis, what we did see was that there was, again, that suppressed immune response. And deconvolution of cell types allowed us to see that there was clearly the macrophage phenotype that was driving a suppressive response that was shared amongst African-American and Native African. So just to sort of recap, that inflammatory response was only shared within our African American, but that suppressive immunity, again, which correlated with our serum analysis, was shared amongst our West Africa as well as our African American men. To investigate this further, we went and performed single cell analysis, and you can absolutely see an influx of myeloid cells within the tumor. You see them spatially located within the endothelial cells, suggesting there's some type of either adherence in or extravasation or trafficking through the vascular system into the tumor. That's just not there in our European samples that we sequence. And if you start to look at the expression of the marker expression, you sort of see in these myeloid cells those same sort of markers that we saw in our serum analysis, those interferon, those sort of suppressor response. And we also saw drivers of those response, which is STAT1 and STAT3. And what does that mean to the proteomic environment? I'll show you some data later, but, there are many reports that show that this high level of suppressive macrophages is associated with a lack of T cell response. Prostate cancers are typically cold. What we saw was that in conjunction with this, in correlation with this in myeloid infiltration, if you stain for the T cell infiltration in these in our African American cohort, we absolutely see T cell infiltration, although exhausted as you would expect it, right? And that correlates with increased PD-1 and PD-L1. As you would expect, and most people have published, and why immunotherapy has largely failed in European cohorts, we did not see that in our European patients, right? We really just do not see that sort of infiltration of immunity in the European, which in our prostate cancer cohort, which is typically associated with cold tumors. And then, just to sort of start to understand, and again, we're very early teasing this out, because in this cohort, we also did multi-omic analysis. So not only did we did single cell sequencing, but we did attack sequencing simultaneously. We were able to find that transcription factors that are regulated this sort of cytokine response seem to be driven by the tumor cell. So if we start to look at clusters of within that population that I was showing in single cell population that overlay with chromatin remodeling in our African ancestry individuals, they all associated with this sort of STAT, immune response. And that seems to be the tumor cell driving that influence, that the cytokines that influence possibly that myeloid infiltration. Again, this is very early on. We have some more analyses to go to sort of define this, but we wanted to share that this seems to be driven by the tumor, and the microenvironment is responding to that. To sort of capture this even more, as we move to Johns Hopkins, actually Keeman, who is an assistant professor here, developed something known as CODA. I thought this was amazingly in a system where we could start to visualize in a three-dimensional format, a modeling system, the infiltration of these immune cell types, particularly across, typical H&E slides. And so what actually was able to do, and she's only previously done this in pancreatic tumors, was able to take serial sections. So we taken an H&E block, we do serial sections. Right now we're at about 100 serial sections, and we're able to look at the tumor in a three-dimensional format, and then utilizing the machine algorithm, AI, machine learning process, you're able to pathologically identify immune vascular tumor cells and or benign cohorts, and then be able to look at that in a three-dimensional model throughout the tissue sections. And so this is just a, as I said, it's almost 100 different slides, right? So this is sort of a top to bottom Z-stack analysis of this. And this is where Ezra Barbarian, our pathologist, went in and looked at each one of these from the H&E, and then assigned this, utilizing the AR machine learning process, assign the individual cell types, and you see that color coded in this images on the right. As you can imagine, this is a European ancestry individuals. We don't see a lot of macrophage and or influx of immune cells. And so if you sort of take that in a three-dimensional format, you can start to, appreciate this even further. You can sort of 3D render this, right? And then you can think about this assigning each one of these cell types a color and sort of visualizing this. And so early in our analysis, what we were able to see is very clearly that European ancestry individual that, I just showed you from the three-dimensional format really didn't show a lot of infiltration. This correlated with our single cell sequencing immune cell infiltration. This correlated with our single cell sequencing data, our bulk sequencing data, as well as our COIA multiplex analysis. But if you can sort of look at our African-American patients, it's robustly filled with immune infiltration. And so we start to see that co-localization within the tumor mass, largely in benign areas, but you see the infiltration around the tumor. And then you can start to appreciate, wow, this robust, inflammatory immune response that we're seeing it from, again, the multi-omics. You can start to visualize that across the tumor types. Across the entire section of the tumor. So, as we started to dig even deeper, right, is this phenomenon associated with just with prostate cancer, or is it across, is it really an ancestry associated response? So again, multiple reports have shown that, it's not just in prostate cancer, right? If you look at African-American women and breast cancer, and this is a really nice review, some of the major differences is this sort of interferon M2, M1 response, right? You sort of see, and you don't get this sort of, active immunity. You get Tregs, which are sort of T regulatory response, and then this upregulation of M2. So our group wanted to, again, being initially a prostate group, our group wanted to further define this in breast cancer. And so we started to ask the question, what was associated with androgen receptor in these patients? And so we started to look at AR positive versus AR negative breast cancer patients. And what we found is that actually African-American women are largely associated with this AR negative phenotype. So over 81% of African-American women sort of have this sort of loss of AR. And if you look at what that contribution, I'm happy to go through, I'm gonna rapidly go through this data because we published a number of papers so we can as well. But what we're able to find is that particularly what we're so-called triple negative is really quadruple negative. And African-American women largely have loss of this AR, right? And this loss of AR is associated with not only just a differential gene expression is associated with a presentation of these women at earlier stage disease. And it's also associated with a basal phenotype, which we know is associated with the typical triple negative. And as well, also this, if you start to do subtyping of these patients, we see this immunomodulatory sort of basal phenotype that's persistent in the lack of AR in African-American women. And again, just to summarize this, we see upregulation of immune cell phenotypes such as CTLA4, PD1, PD-L1, as well as B7-H3. And again, started to dichotomize these patients as based on their ancestry. We ran a transcriptome analysis and started to look at, this is a classic example if you start to look at differential expression by self-reported race. You start to see some differences, but if you start to run genomic qualified ancestry, you can start to appreciate a separation of gene expression profiles in a European versus African-American, right? Or African ancestry individuals. And if you really start to notice that these European, genomic ancestry individuals that sort of start to express differentially genes similar to African-Americans, they actually phylogenetically cluster with our African-Americans, right? And so again, this ancestry sort of matters to the biology. We went on to show in a subsequent manuscript, collaboration with Melissa Davis, and got the cover of Cancer Discovery, that this is not just associated with African-American, it's associated with West African. So if you start to look at, the contribution of ancestry through Ghanaian individuals, and 2 cohorts from Detroit and Alabama. Again, I was at Tuskegee and UAB before I moved to the Johns Hopkins. You can start to see that these patients are predominantly of West African ancestry. What really became interesting is that if you look at patients that we collected from Ethiopia, that was quite different, right? So these Ethiopian individuals actually have 50% European, 50% West African ancestry. And it's really interesting how this plays out in their biology. So again, started to look at the biology of individuals. We were able to clearly see a gene expression profile that was Ethiopian only. There was a shared genetic profile, which was largely West African from African-American men and Ghanaian women. There was a clear genetic gene expression profile only in African-American, and then there was a shared African profile that was in Ethiopian and Ghanaian women. And if you start to look at, the contribution of African ancestry or European ancestry and self-reported race, right? Because both of them matters, right? Self-reported race matters, ancestry matters. What we were able to tease out was that the ancestry genes assumed to be associated with this immune response, right? Whether it was West African ancestry, and it's really interesting that the West African ancestry and the East African ancestry actually go in different responses, although both were associated with African ancestry. They sort of triggered. So if you think about the Ethiopian, which is highlighted in the yellow here, is more pro-tumor, right? If you look at the West African area, it's more suppressive environment. But what's also really interesting, if you look at what we call race-associated genes, these are genes that did not correlate with West African ancestry. They are sort of depictive of comorbidities, right? Cardiovascular disease, obesity, and diabetes. So, this was really interesting, right? So we've started to set up that there's a clear phenotype, there's a clear suppressive phenotype across breast and prostate cancer, but that didn't tell us the mechanism, right? We're molecular biologists, and we want to start to understand, are there targetable mechanisms in these patients that or pathways that can be exploited for therapeutic targeting? So our lab had been focused for many years on a protein called Kaizo. Kaizo was one of the very first ancestry-associated biomarkers for breast and prostate cancer. And Jackie Jones, who is now at Troy University, was able to show that this was upregulated in pancreatic patients, breast cancer patients. And you could start to see, regardless of the expression of Kaizo in primary tumors, in METs, you start to see it increase. And then in African-American women, absolutely have more Kaizo compared to European. Julia Daniels, who was actually initially coined this, was able to follow up our studies and showed that not only is this upregulated in African-American women, if you look at women from the Caribbean or Barbados or even Nigerian, it's even higher, right? So this was increase in prostate cancer tumor patients as well. Again, Jackie was able to show that, and then also in pancreatic as well. Really interested in the pancreatic cancer, it was really that we saw a clear gender difference, right? Well, so, Kaizo was upregulated in Afro-American women higher than men, but as men progress to sort of high-grade disease, you start to see them sort of catch up, but there was a baseline overexpression or elevated expression of Kaizo in women with pancreatic cancer compared to men. And just for the individuals, we have extensively worked on Kaizo and tumor cells, and this is a review. Julia Daniels' lab largely shows that Kaizo's, particularly gene expression, is regulated through TGF-beta signaling. Our lab has been largely focused on EGF receptor signaling, but the net result is that Kaizo seems to be regulating multiple would be at the center of multiple pathways that's regulating NL metastases. And if you start to look at the processes, it's involved in a myriad of processes, just like all the other EMT-associated genes, such as chemoresistance, EMT, response to chemotherapy as well. But what was not known was his role in immunity, right? And so as we sort of, still were exploring the multiple roles of Kaizo in disease, in collaboration with Kevin Gardner, we published in Nature Communications some time back, that if we started to look at Kaizo expression in, African-American women breast cancer patients, were they associated with an infiltration of immune response? And they absolutely were. So if you look at high Kaizo expression tumors, what we see during nearest neighbor analysis is that you start to see in sort of that niche neighborhood, CD8 positive cells that are co-expressed with PD-L1. And then you can sort of appreciate that nearest neighbor analysis graph on the right. And that was, some of the most associated immune cells that were clustering around the neighborhoods of these high Kaizo expression tumors. And so since that was, this sort of neighborhood response, we were sort of, envisioning that there could be some type of secreted cytokine crosstalk. And so, we wanted to look at the exposure of these individuals through exomes analysis. And so we assayed exomes, which are these small microvesicles that were secreted in African-American versus European. And we absolutely saw that, this increase in JAK-STAT signaling and FKB was upregulated in our African-American tumor exosomes compared to European. And if we wanted to look at our 231 breast cancer model, we saw this also, in our cell lines. So knocking out Kaizo actually completely altered the immune response in our tumor in our tumor cell line. And if you overlay these differentially expressed genes from our, knockout model with the differentially expressed genes in our African American, what we got was a possible set of kaiser-regulated genes, particularly TGF-beta, which was really great for us because we know TGF-beta regulates kaiser, but we also saw macrophage markers such as CD47 and this interferon response, again, that was largely been associated with, these sort of high-level, studies that we saw in breast and prostate tumors. And then if we were able to knock out and inject these into, mice, what we largely saw, and this is athonic new mice, by the way, so we don't see T cell response, but we also were able to assay macrophage influence. Depleted Kaiser cells were able to obviously have a decreased tumor growth, as you would expect. But when we largely look at the microenvironment, we largely saw a complete reversal, right? It's clearly classic that, these breast cancer cells present with, it's particularly in the 231 model, increase in, suppressive macrophages such as TD206 higher and in CD163. We saw in the Kaiser knockout or the depleted Kaiser cells, we saw more of an M1 response, which was high CD8, 80 and CD86 compared to, the sort of suppressive in the control cells. To sort of validate that this was sort of real, right? We did, some co-culture experiments where we took exosomes from our, sort of knockout and control models, and we applied it to, non-metastatic MCF7 cells. And again, just applying the exosomes from our knockout and our control, we're able to see upregulated gene response, right? So in the MCF7 cells, they actually became, and I'm not showing the data here, they became more invasive. They underwent EMT, and we see increased Kaiser as well as CD47 just by sort of this transfer of, these Kaiser high versus Kaiser low cells. And we saw almost a similar effect on THP1 macrophages as well, right? And so again, modulating the tumor cell and being able to modulate the microenvironment, Kaiser seems to be playing a role on what's secreted, right, and influencing the activity of these. And when we ran a phagocytosis assay, we actually able to see that, right? So conditioned media or cells from Kaiser depleted cells were being phagocytosed more by our macrophages than this SH Kaiser, which, we also saw upregulated CD47, was largely sort of resistant to what we think about phagocytic mechanisms. And as we start to think about, and again, this is very early on in the data, we were really inquiring, is there something about Kaiser regulating gene expression that's differently being secreted in the microenvironment? And while I don't have the specific assets now, we're running a host of RIME and cut and run data that we're actually analyzing now, we do have a hint. So we published a paper largely where Alexandra Harris at NCI, who was a postdoc, and Brittany Lloyd when she was a postdoc at NCI, and she's now a junior faculty here at Johns Hopkins. What they asked was, utilizing our African American model, right, of breast cancer in African American cell lines versus European, what was different at regions of open versus closed chromatin? And what they saw was that there was a very difference in our African American cohort at open regions of chromatin in our African American. And if you start to look at what genes were associated with regions of open chromatin in African ancestry, our Kaiser genes, as well as other methyl binding proteins, as well as this whole methyl complex, was sitting at regions of open chromatin that we absolutely did not see in our European. And as I said, now we're sort of fleshing that out to the sort of, gene expression signatures that could be regulated by Kaiser, and I hope to present some of that data later. But, just sort of take home, sort of this part of the story is that we aren't truly seeing that there are biological differences in these transcriptional regulation, and possibly this is regulating what we're calling, the secretome, influencing this sort of suppressive microenvironment and or immune escape in African American, which is not sort of happening in a low Kaiser expression tumor. Now, we were, again, very excited that this sort of myeloid sort of phenotype was, sort of persistent in African-American patients. And while Kaiser is a transcription factor, and the biology is very fascinating, and I think we're just cracking open, the sort of biology associated with this, it's very hard to target a transcription factor. And so in collaboration with Jelani, we started to think about going after, from a therapeutic perspective, just directly start targeting the macrophages. And so we published this paper in Science Translational Medicine some time back, where we developed a peptide inhibitor to CD206 with canonical macrophage marker, many of you guys have used in your lab. Well, we sort of see, targeting CD206 causes this sort of conformational change, particularly targeting the CRD5 domain, sort of causes this conformational change in the CD206 receptor that it causes immediate repolarization of M2 macrophages into sort of the N1 response. And when I say immediate, is really sort of almost de novo. So as early as 30 minutes, treating sort of M2 polarized macrophages. And again, I know we don't like the M2, but I'm, sort of phenotype, but I'm going to use that just to sort of talk about our CD206 positive population that we're sort of assaying here. You sort of see this sort of conversion, and that sort of repolarization occurs over a 24-hour period. We don't have time to go into the entire mechanism of how this, but this is sort of a summary where we're able to show that CD206 actually has a signaling, prior to these studies, it was just a canonical marker, but we were able to show that engagement with this peptide creates a signaling cascade that starts with recruitment of CD47, activation of RAC1, CD47 RAC1 pathway, ultimately activating PAC1 as an intracellular molecule, ending up with NF-kappa B, and secretion of TNF-alpha in a sort of acute response. That secretion of TNF-alpha happens almost immediately. We think that's responsible for the sort of macrophage repolarization, and sort of the secretion of the TNF-alpha through an autocrine mechanism is responsible for the death. Right. And so you sort of see this sort of immediate repolarization while we think it's happening almost de novo. And you sort of see this death of a decrease of CD206 positive sort of happening later. But if you actually gate for these, CD206 positive population, again, you see this robust increase of TNF alpha, but you also see innate immune response genes such as IL-1 beta, IL-1 beta and IL-12 beta, as well as you see a committed decrease of PD-1 and PD-L1, as well as SERPA alpha, and which we know is again a ligand for CD47, a decrease in these macrophages. As we started to, I've moved to, Johns Hopkins, and there's no secret that, and Jelani said that I'm obviously the John Lewis endowed professor, and we know John Lewis passed the pancreatic cancer. We've asked, is this sort of model of disparities that we saw in our breast and prostate cancer patients extended to PDACs? And it is. So if you start to look at CD206 expression, even in healthy individuals, we see increased CD206 expression in M2 polarized macrophages from healthy individuals. And if you think about treating macrophage, sort of monocytes, with serum from African Americans or serum from European PDACs, you can sort of see that even more exacerbated, right? And we were able to take this and sort of, RNA-seq these individuals. Again, these are very early on. We're clearly able to see, in African-American serum-treated macrophages, a differential sort of mixed M1, M2 response to sort of increase in this IL-10, all these sort of macrophage, these sort of cytokines that we saw in our sort of suppressive immunity response in our prostate and breast cancer, we see absolutely at the gene expression level and the sort of African-American tumor response, which we do not see in the serum treated from European Americans and its macrophages. But just back to, sort of our M2 peptide treated data, you can start to see even, treating with our peptide has an effect, and these are P16 KRAS model, but completely clearly in combination with gemcitabine, and I'll show you along, we clearly see that there's an additive effect of, with synergizing with our peptide, and this, associated with, an overall increase in survival. Not only were we see this in gem models of pancreatic cancer, we already see this in PDX. So if you look at high CD206 expressing PDX models, clearly respond to our peptide. What we do not see if we chose PDX models from low expressing CDX patients. And then, we successfully asked the question, does this synergize with IO therapy? And absolutely, again, PDACs are largely unresponsive to immune therapy. And you can see that by this sort of, varied response to PD-L1 as far as the, single therapy. But in combination with our peptide, we sort of get this sort of overall induced response to PD-L1 therapy. And then because we think, this macrophage response is also dependent on the T cell response, if you actually, treat with a CD8, an anti-CD8 antibody, you can sort of mitigate this sort of gained, response of PD-L1 as far as, during RP1A2, which is our macrophage cell therapy. So we're starting to understand that this, that this macrophage is associated with this T cell response, and we can see that very clearly from this flow cytometry data. That again, decreasing CD206. Decreases CD not only M1, but you see an overall decrease, modest decrease in myeloid suppressive cells, but that also is associated with increased CD8 cells, as well as an decrease in Tregs. And you see that not only at the by flow cytometry, but we can see them infiltrating into the tumor parenchyma, as well as you look at this, and this is from P16, and we also did this committedly expression in our KRAS model. And just because, we wanted to sort of look at not only what was happening as far as our immune response, and because we had this sort of EMT Kaizo response that we were seeing early on in our breast and prostate cancer, we asked that same question, and absolutely we saw that our peptide reversed, EMT markers such as ecoherin, vimentin, and obviously we saw a decrease in Kaizo expression when we treated or depleted macrophages, particularly CD206 in these sort of PDAC patients. And not only do we able to see just a decrease in, we also saw actively suppress in sort of immunity. And so here is, again, these are macrophages, and these are calcium stained tumor cells, and you can actually see how the macrophages start to engulf or phagocytose these tumor cells. And then if you look at this in our tumor types, you can actually see more of these foamy type macrophages, and whether that's for an H&E or for electron microscopy, you can sort of see this engulfment in our peptide-treated tumors compared to control. And so not only do we see this associated with PDAC models in collaboration with Roger Lowe at UCLA, he was very interested, very similar to that we showed, from our breast and prostate cancer patients. Looking at targeting MAP kinase pathways, we talked about Kaizo being downstream of EGF receptor signaling. We know that's associated with MAP kinase. What Roger was looking for was that, what he was seeing in his patient tumors and then modeling that in vivo is that you start to see, MEK inhibitors kind of have this sort of therapeutic resistant response, not only in human, and that start to bear itself out in multiple models, particularly in melanoma, as well as pancreatic models, and syngeneic models in vivo as well. And when he started to look at, as tumors became resistant, what were the cell types that were associated with? Yes, we see this sort of infiltration of the sort of TAM associated phenotype being, some of these immune cell types that started to be associated with when tumors started to recur. And so just phenotyping those even further, what Roger was able to show in collaboration with us was that, we see this sort of interferon macrophage response. And then if we were able to utilize our peptide as far as, pre-treating these and then start in what we call in these sort of lead-in studies, and then start with our MEK inhibitor and then subsequent PD-L1. Again, these mice were largely unresponsive to the PD-L1 therapy. You can sort of prevent this response. And so, again, that's what you see, this sort of targeted macrophage therapy here. And at the bottom is our sort of pre-treated peptide individual mice studies where we sort of, prevented that macrophage, at least this M2 CD206 positive phenotype, and then the MEK inhibitors as well as PDL was actually able to do what it's supposed to do and target and decrease tumor volume. And then lastly, I'll just go through what I think, sort of what we think is utility of this. Not only were we able to see associated with cancer types, we published a paper, and then we think, as we're developing our clinical strategy, this is probably going to be, one of the first therapeutic targets that we're going to, think about our peptide as far as a clinical trial. But, we see this decrease in lung fibrosis as well. We did publish that in our original STM manuscript, but we actually followed this up in the second study, where again, we saw targeting the CD206 macrophage, particularly the infiltrating ones into the lung parenchyma after bleomycin, really decreased lung fibrosis. And then subsequently, it's really important for us to think about companion diagnostics, particularly, macrophage therapies has been kind of, except for this sort of recent study, the CD47 that was more favorable from, I think, that, VinBio, Axion therapy that, data just came out, largely, has been kind of beat up as far as the therapeutic space. So we thought it was really incumbent to understand, when should we start treating this? And so in collaboration with Candace Parker, who is a graduate student at UAB, who, is our co-mentor, she's now moved on with Memorial Sloan Kettering, and Susie Lappy's lab, who's a molecular imaging, we tagged our peptide to a radio imaging agent. In this was case of gallium-68. And we were able to demonstrate that we are able to effectively visualize when these macrophages sort of start to infiltrate. And you can sort of see that in a CT26 colon model, which has really high expressing CD206 cells, is really duplicative of, this sort of infiltrating this particular CD206 response. And in a low CD206 expression 4T1 model, you actually don't see this low level of sensitivity. Again, we published this early last year, and we think this is really important. We just finished up data that is showing the same sort of data in lung fibrosis. So we think that, this is really, really important as far as understanding when a macrophage, particularly a CD206 targeting macrophage China therapeutic, should be delivered. And now I'll just close what I think is sort of the utility of this. We performed a CD206 imaging study to understand, how can we sort of utilize an imaging agent to best capture, when this macrophage biology is important in tumorigenesis. And so again, utilizing our CT26 colon cancer model, and over a multi-day period, we started to image, sort of this, response to multiple IO therapies. And in this case, it was CTLA-4 is in combination with PD-1. And so again, what you can sort of appreciate over this sort of tumor volume, right? Early tumorigenesis, day 3, as many of us have shown, right? If you combine this sort of studies, you get sort of this sort of clearly responsive tumors that sort of decrease tumor volume and get this sort of unresponse. What we were able to see through our imaging agent is that these sort of unresponsive tumors were able to uptake more of our gallium or radio-labeled CD206 peptide compared to the ones that were kind of responsive, which it suggests, very similar to our studies that we completed in our with Roger Lowe, is that, that we were able to see that this macrophage sort of response, particularly even to T-cell therapy, was able to be predictive of this sort of non-responsive patients, particularly to checkpoint therapy. But this is where it got interesting. Remember, microimaging is utilizing really very low dosages, right? So we're talking about very low. In this case, we're talking about 0.5 mg per kg dosages. And so what Susie and was able to do, and Candace was able to do during her graduate studies, is she said, well, if we were able to image that, and we know that our peptide has some, synergistic activity, what if we gave a second imaging dose, right? So we started off with an early imaging just to predict the non-responsive tumors, and then we followed it up later with a day 9. And so what was that impact on, overall response? And so the nuts and bolts of this is that even at micro dosaging, right, very low dosaging, we're talking about 0.5 mg per kg, we were able to see a large number of those non-responsive tumors become responsive. So just eliminating these macrophages, I'm not showing you all the data here. We saw macrophage engagement of our peptide as early as 45 minutes, probably between some 30 and 45 minutes. But engagement of these macrophages, even early on, allowed for this, sort of IO type combination therapies to be more effective, even at low dosages, even in a sort of an imaging study. And so I'll just end that, and take time for questions. Is that we do disparities in our lab, but we really think it's a personalized approach. And so I'm really interested and very happy as we're progressing to our particularly our CD206 peptides to our clinic. Gelati just got an R01, really studying how this will be associated with our peptide will be associated with targeting prostate tumors. But, I just really like to end with, I think, a study done in prostate tumors, particularly exploiting this sort of an inflammatory immune response. So Oliver Sorter, who is a colleague and an oncologist, utilizing a larger biology that, us in other labs have shown for African-American men having this immunomodulatory response, that if you think about prostate cancer tumorigenesis and, you think about androgen deprivation therapy being the mainstay, and then you start to move on to those patients who, relapse into chemotherapy. And he said that, based on the biology of these individuals, you should really think about applying immunotherapy. Again, that's largely been ineffective in European populations for prostate cancer. What he was able to see in a phase 2 clinical trial was that African Americans actually responded twofold better and had much more robust outcome than European patients. Again, based on the biology that, us and others have been able to see associated with African American men. And so I'll just sort of end there because, these are the people who do all the work. I get to talk about this, both through, in venues like this as well as nationally. But these are the individuals over the years that have been through our lab and now have moved on. And as well as our West African, collaborators who have come to our lab, been trained, and now are back in their country, being able to do a large amount of, histochemistry, immunological assays. And this is sort of the group that we're really proud of. And here are all the collaborators that and the funding that's contributed to being able to get this work done over the years. And so I'll stop there and take any questions.
Great. Awesome talk, Clayton. Lots of exciting data there. We'll take questions. I don't see any in the chat.
Hi, this is Judy. I sorry I came in late. I just want to thank you so much for giving this amazing talk. There's so many wonderful nuggets of information in your talk that I have a billion questions, but I wanted to ask whether there's a difference in the African American population in different locations in the United States. Have you looked widely across the South and the West, and whether you see any differences in the responses or the immune phenotype that you're studying?
Yeah, so we haven't, right? So if you look at some of our Alabama patients, again, I was in Alabama before I came. That last cohort that I showed you was a Maryland cohort. If you look at the Caramos data from Michigan, what we're seeing, John Carpenter's group in California has shown the same thing. It's pretty much the same. It's pretty much this sort of inflammatory innate immune response. And, that question we didn't ask across the US, but we kind of asked, was it in West Africa? And that's how we kind of went to Africa, but we clearly see it. Now, I will say we just got a Cancer Grand Challenge Award funded, which we're going to recruit 40,000 individuals across the globe, right? And ask that same question, right? So we got, this Cancer Grand Challenge, it was 25 million, we'll recruit 40,000. 15,000 will be prostate cancer, but it'll be pancreatic as breast as well. And so we can more robustly answer that question, but just anecdotally, no, we don't see any difference.
Yeah, that's super. So
hey, Clayton.
Oh, go ahead. Sorry.
No, I was just going to say, and follow up to that question, the real interesting question would be looking at people from Eastern Africa and Southern Africa, and then comparing them to people from Northwestern Sub-Saharan Africa. And then you can try to figure out the environmental causes for this difference.
And I'm sorry I didn't put up the slide, but that's exactly the purpose of the Cancer Grand Challenge. So we will be in 14 different countries. East Africa, based on that data. So that you can see all, I tell people all the time, grants are reimbursement, right? So all of that data that we use, we were able to go and say, hey, there's a clear difference in East Africa. There's a clear difference in South Africa. There's a clear difference in West Africa. What is that contributing to the African ancestry and African Americans? And then, so we're doing exposures with Gary Miller through Columbia University. So we'll be doing exposomics. So we'll be able to tell the exposure of all these individuals compared to genomics, and then we'll be molecular phenotyping through Akoya and Xeon and Xenium and all of these other fancy things that I just showed you there. We'll be phenotyping those patients as well, and then so hopefully we'll be able to decipher this out. It's going to take us probably another 5 to 6 years to do this, but we're starting now to sort of dig through and that's exactly the point of the question. So can we contribute, what pieces of ancestry contribute to what phenotypes and or exposures, overlaying that, as well as social determinant factors, because we'll be collecting social determinants in those individuals as well, and then see if we can really start to tease this out.
So can I ask more about the Kaizo? Have you
Yeah.
figured out what really it drives? I guess does it drive TGF beta or have you, or is it something like IL-4 or do you know yet?
Yeah, so right now it seems to be a feed forward loop, right? So clearly TGF beta. So and I didn't show the data there, but you know, we treat with TGF beta or EGF, Kaiser goes through the roof. Okay. It just,
Oh, I see.
it goes through the roof.
Yeah.
Now, what we also found when we did our chip experiments is also regulating the ligand for, these ligands. So if you look at the ligands, and again, I didn't show all the data, Kaiser is directly binding to microRNAs that are intermediary responsive to that. So, we have papers that show that there's a micro mediator of this. So it's binding to the 200 family, 145. So all of these, so until we got into the immune response, we were really associated with that more EMT, epithelial mesenchymal transition sort of response. And we teased all of that out. And yes, there seems to be this feed forward loop where Kaiser's driving that ligand, because we know in order for metastases to occur, it has to be disorder autocrine. Like cancer cells have to be able to survive. John Cordelis's work, like 2 macrophages and secreting EGF through the, through the vasculature that sort of support. So we think Kaiser is a mediator of that growth factor driven response that sort of helping tumor cells survive through metastases.
That's great.
Hey, Clayton, what do you think the major driver of the inflammation is in the tumors in African Americans that Oliver Sartor is seeing that you don't see in other populations? Is it more than Kaizo? What else could it be? It seems like potentially myeloid cells get to tumors and become inflammatory macrophages or some type of MDSC phenotype that's very different in other populations. What do you think might be driving that?
So our TAC-seq data says that it's the STAT family that's coming to the top.
Okay.
So the STAT gene family, and it's from the tumor cell. So we didn't see that clear African-American from again, this is early on, Israel very well, Jesse, July, she's in the lab finishing up these analyses. But what is clearly from our data so far is that when we start looking at where these sort of transcriptional factors, are located, and because we did multi-omes so we could determine gene expression, 2 chromatin remodeling in the same cell type, we see those STAT genes locate to regions of open chromin really in the African American tumor cells, and we see the ligands really expressed in the myeloid cells. We're doing pairwise analysis right now so we can see, which cytokines that these STATs are directly regulating and which receptors on the sort of myeloid. So that data is coming, but it seems to be driven. And I didn't show the data either. We actually macrodissected out some tumor sample cells. So we, as much as we can, almost not laser capture, but macrodissect out. So we tried to enrich any other immune cell response, and we still see the inflammatory immune response, even in the macrodissected tumors, compared to the macro. So, we see it seems to be still driven by the tumor cell.
Gotcha.
So I noticed that the Kaiser, sorry, so a lot of noise here. I hope you don't hear it. But I noticed that the Kaiser is driving STAT3 expression or activation. Is that right?
We're not sure. So we kind of looked at Kaizo in our breast cancer set. Obviously there, in the breast set, we saw a little bit differential. So there, we saw some 8 response. It's very clear this innate interferon response was the major gene STAT in our prostate tumors. So we clearly see, this sort of STAT driving prostate tumors. Actually, I had her look for Kaizo, and while we think Kaizo is important in prostate cancer, it didn't come up as the most significant driver of disease from our TAC-seq that when we co-localized it. We're not saying that Kaizo doesn't play, but when we looked at the top genes, it seemed to be, in our prostate tumor, it seemed to be STAT genes. In our breast cancer, this sort of macrophage response seems to be associated with, this sort of Kaizo response. And so we're still teasing that out between the different models. But there's probably some redundancy in that too. So, you know how cells are. There's Mother Nature's redundant.
So I had one more question. Sorry to dominate, but
Yes.
so the beautiful CD206 peptide, reagent, and you're doing clinical trials. Are you doing that are you doing that widely across ancestries? So African American, you know, European, or are you focusing it mainly on a certain population? And because
So
it
we're
looks
finishing up
it
some
would
of
work
the
in
IND
most.
studies. Yeah, we haven't finished. We're finishing up some of the IND studies, so we haven't done our patient stratification yet. But a
Yeah.
larger part of this work is going to suggest that we need to over recruit for African American. Now,
Yeah.
again, we utilize, typical, endpoint or entry criteria would be like CD206 positive population.
Yeah.
But that's also why we developed this companion diagnostic, right? So we really like this radio imaging. We're sort of finishing in parallel. So the radio label peptide is actually caught up to the therapeutic as far as development because it's kind of hot. This radiotherapy is pretty hot now. You guys know all about the PMSA and prostate, data. This is sort of leading that. So, and also because of the large number, and I'm happy to talk about, these other macrophage therapies not doing so well in cancer, right? It sort of, prompted us to say, hey, we need to go to imaging while we're sort of making sure our biology and patient, sort of stratification is good, as well as sort of look at our fibrotic tumors. And that's why we sort of pivoted from a, sort of entry part to our fibrosis sort of patients versus the cancer patients while we're sort of figuring that out. So that's been our strategy. We did, spin out for, a small startup. And so that's been the strategy from the startups perspective. Yeah.
Yeah. Okay. Well, I do have one more question, if I may. So we have noticed in lung cancer models, mouse models, though, that there's a huge upregulation of CD206 on macrophages in lung cancers. Is there an association of lung cancer outcomes in African American or West African populations? Or is
I haven't really looked. That's really important that you said that too, because we haven't tried lung cancer and we think our peptide does very good in lung fibrosis. So
yeah.
we have really great data in lung fibrotic diseases, so it clearly gets to the lung. But no, we haven't really looked in, as far as the disparate outcome. We do know there's a response difference, right? So we do know African American and Asian, not necessarily African American, but like people of Asian ancestry don't respond to the TKIs as much as, right, because of the, difference in the mutation of the EGF receptor expression. So we do see not just a disparity in African American, but there's a clear disparate therapeutic response because of that mutation that was held, in people of Asian ancestry. So, we do African American ancestry, but we would think ancestry is a fundamental response to all populations as far as understanding how we should be treated and what drugs should we use in treatment, particularly in our targeted therapy settings.
Yeah, that's great. Well, if you're interested in doing a trial, across coast, I know our cancer center would love to work with you on that. So
absolutely, thank you.
Yeah.
All right. Well, if there are no other questions, Dr. Yates, thanks so much for the compelling seminar. We all learned a lot on this call, even those who didn't have questions.
Thank you for the invitation. Thank you and Judah for basically inviting me. So I thank you guys very much.
Yeah, thank you.
Shelly's there too. Hey, I remember that a long time ago. How you doing, Dr.
Yeah, thanks again.
Take care.
Yeah, you too. Thank you.
Bye-bye.
Jelani.